Thanks everyone for joining this week's seminar, the online edition. So it's our great pleasure to have Sabrina McNew from Cornell joining us. And today's actually going to be a double feature because we're going to have Teresa introducing the speaker. So Teresa is no longer at the Institute, but she was the one who invited Sabrina. So we're really happy to have her back for this week's seminar.
Yeah, thanks, Chris. Well, hi, everyone. I'm really happy to be back at EBD, even if it's just virtually. And I want to thank you and also Agus for letting me introduce my friend, Sabrina. She's a postdoc working on bird disease ecology in the Lab of Ornithology at Cornell University in New York State in the USA. Before that, she did her undergrad in biology at Pomona College in Claremont, California. And then she did her PhD at the University of Utah. But I'm sure she'll tell you more about her. So Sabrina, whenever you're ready, the virtual stage is yours.
Hi, good morning and good afternoon to all of you. And thank you so much for that introduction, Tere. I'm so pleased to be here. Of course, I wish that I were in Seville with all of you, but these virtual introductions are a great way to meet new colleagues. And so I'm really happy about it. I wanted today to start by telling you a little bit about myself and how I got here, because I know I'm new to all of you. And so, like Tere said, I'm from the American Southwest. I grew up in New Mexico. I went to university in California, ultimately did my PhD in Utah. And since I received my PhD, I've been working as a postdoctoral associate at the Cornell Lab of Ornithology on the other side of the country in upstate New York. But what has really shaped me as a scientist and as a person has been the opportunity to work in the neotropics, primarily in Latin America. And so starting in just after university, I got the opportunity to do fieldwork in mainland Ecuador. Later, I worked some in the Peruvian Andes. And then since graduate school, a lot of my research has been based in the Galapagos Islands, which are off of the coast of mainland Ecuador. And in each of these places, these are incredible biodiversity hotspots and wonderful places to work. And I started with an interest in birds and ecology and animal behavior. And over time, that shifted a little bit into a focus in species interactions, particularly interactions between hosts and parasites. And so in each of these amazing places, as incredible as the birds are, I often spend more time looking inside them or around them at the other species that they interact with. And I'm interested in these interactions for one particular reason, and it's a very simple one, and that's that parasites are everywhere. So just as an example to get us going, consider this pigeon, which is standing here preening, something that birds spend a lot of their time doing. And I might ask you, why does the bird do this behavior? What do pigeons preen for? And some of you might be thinking about how gross pigeons are, which is sort of true, but again, all birds spend a lot of their time preening. Feathers are essential for thermoregulation. They can play a role in sexual selection, but preening also serves one particular important function, and that's the control of ectoparasites. And so if we could zoom into this bird, we might see buried within its plumage a hippobossid fly. This is an insect that lives in the plumage of the birds. It feeds on the bird's blood, and it's an obligate parasite of this pigeon. And if we look closely at that fly, we might zoom in even farther and see yet more parasites. These are feather lice, more obligate parasites of birds. They feed on the feathers of the bird, but occasionally we'll hitch a ride on these flies to get from host to host. And then if we look closely at that louse with a high-powered microscope, we could zoom in and see some bacteria, perhaps commensal, perhaps parasitic themselves. And my point here is just that the closer we look, the more parasites we see. And here, let me take a moment to specify that when by parasite, I mean an organism that derives resources from their host and also reduces the fitness of their host. And it's this antagonistic, intimate interaction that's so interesting to me because, of course, parasites need a host to survive. And at the same time, the effect that they can have on the fitness of their host imposes selection. It can be an important selective force. And indeed, I believe these interactions can affect anything that we might care to study about in ecology and evolution, from individual traits, such as the preening behavior I showed you in the video, to even entire populations. And the famous example of this is the decline and extinction of many of the beautiful species of Hawaiian honey creepers in the Hawaiian archipelago due to the introduction of avian pox and avian malaria that really decimated those birds. And host parasite interactions can even infect entire ecosystems. And so one case of this is the eradication of rinderpest from the African continent. The eradication of this disease shaped this ecosystem by creating a boom in wildebeest, which changed the grazing in the habitat, leading to lower fires, and over time, a shift in this ecosystem from a more grass-dominated landscape to a more tree-dominated one. And so I'm really fascinated by these interactions because of the myriad effects that they can have. And this big question is the one that I'm interested in. How do host-parasite interactions shape ecology and evolution of both partners? I am interested in this question across scales, spanning from communities to individual outcomes to even individual genes. And today I'd like to tell you about some of my research on the smaller side of this scale. And I will tell you about work that I do in the Galapagos Islands, which are in the Pacific Ocean off of the coast of mainland Ecuador, a really incredible place to work. And I will talk about the effects of emerging parasites on naive hosts of these islands. These islands are a great laboratory for studying how birds respond to a rapidly changing environment and the introduction of novel threats. I'll return to this outline to keep us oriented, and I'll give you three brief vignettes of some of the work I've been talking about, sort of hot off the press, a project that I've been doing during my postdoc, studying the effects of parasites on host cellular function. Then I'll tell you about some work I completed previously, investigating the effects of parasites on host fitness. And then finally, I'll try and integrate this research with a work I do in the community, promoting education and training to support the management and conservation on the Galapagos Islands. So just jumping into part one, let me tell you a little bit more about the Galapagos Islands. They are a volcanic archipelago, meaning that they've never been connected to the mainland. They're quite remote in the Pacific. Of course, they were famously visited by Darwin, who used his explorations of the islands and his observations of their geology and unique inhabitants to inform his ideas about evolution. But they're special for another reason as well, and that's that they were long insulated from human disturbance. And so the Galapagos were one of the last Pacific archipelagos to be settled by humans, and that means that they were long insulated from the sorts of death and destruction that we typically bring. On the ground, they look something like this. They're a harsh but a beautiful place to live if you're a little bird or another organism. And even though these islands have been relatively well insulated from human disturbance, there's been a huge rise in development and tourism in the last couple of decades, and that has presented a lot of new threats to the endemic species of the Galapagos. So for birds, which is what I focus on, these threats include habitat loss, especially in the humid agricultural zones of the islands, new predators such as rats and cats, and of course, introduced parasites and pathogens. The first one that I'd like to tell you about today is called avian pox. This is a disease caused by a pox virus. Infection causes lesions on the feet and face of the birds, and this infection is not necessarily fatal, but it can impede mobility and foraging. Passerines, such as these Darwin's finches, get infected, and even some seabirds like the critically endangered wave albatross. We believe that this virus is introduced to the Galapagos and is spread through direct contact between individuals or potentially through mechanical transmission. So a mosquito landing on one infected bird and then landing on a different one could spread the virus. This virus is a cause for concern in the Galapagos because, like I mentioned, it is associated with the extinction of a number of beautiful Hawaiian honeycreeper species on a different archipelago known for a different radiation of birds. So all of the birds in gray in this picture, all those little faces are species that are now extinct, in large part due to introduced disease. And so in the Galapagos, we haven't yet seen these declines, but we are concerned about this disease. And moreover, we have a question more generally about why is it that some species can survive a plague or in the introduction of a novel pathogen, but others go extinct. And so among these array of honey creepers or in the Galapagos Islands, are certain species more susceptible than others? And why might that be? So to try and address this question, I began a project as a postdoc to investigate the transcriptomic response of Darwin's finches to pox infection. More specifically, I wanted to understand first what genes are activated in response to infection. Second, what are the function of those genes? And I thought by answering this question, I could learn a little bit more about whether the finches are even able to detect this pox virus and whether they respond adaptively. So this work started in the field. We catch birds in mist nets. We can take blood samples, look for signs of infection. And by we, I mean myself and 2 field assistants who I worked with, Hanae and Diana, who live on the Galapagos Islands. And then in the lab, I designed an RNA-seq experiment with the help of our core facility. I focused on 2 species that are common in our study area, the medium ground finch and the vegetarian finch. These species co-occur, but they're not sister species in this radiation. They're from sort of different branches of the Darwin's finch tree. I selected 10 infected and 10 uninfected birds of each species and sequenced total RNA. That gave me a whole lot of data, information about the expression of some 16,000 genes. And I analyzed differences in expression and potential functional relationships of those genes using something called gene set enrichment analysis. So let me tell you a little bit about what happens under the hood here. So some of you may have seen a mosaic plot that looks like this, which tells us information about gene expression. Here, each row is a gene, each column is an individual, and the color tells us how highly that gene was expressed. So say we sort this list in correlation with the phenotype of interest, meaning that at the top, we have all those genes that were more highly expressed in infected birds. At the bottom, all those genes that were more highly expressed in uninfected birds. And then in the middle are the genes that display no correlation with the phenotype. Then I can compare this sorted list to a list of relevant genes whose function I care about. Say, for instance, we know that certain genes are involved in immune function. And if those genes just happen to occur at the top of my sorted list, then we can statistically say that there's a relationship between the phenotype and this particular biological process. So next I need a relevant list of genes that I can compare to my sorted list. And today I'll tell you about hallmark gene sets, which are curated by UCSD and the Broad. And each of these 50 sets contains some 20 to 200 genes that we know are going to be co-expressed and typically involved in a particular biological process. Okay, so moving on to some results. The first thing I'll tell you is that infected birds of both species upregulated expression. So here, just comparing my infected versus uninfected ground finches, I saw that there were a number of genes that were significantly differentially expressed. So this is a volcano plot. On the x-axis, this tells us the difference in expression between infected and uninfected birds. The y-axis tells us how confident we can be that there was a significant difference in expression. And all those points that are labeled are ones that were identified as significantly differentially expressed. And the fact that they're in that upper right-hand quadrant tells me that these genes were turned on in infected birds. So then I can feed all of this information into that GSEA analysis. And indeed, this produced a number of significant hallmark sets. I will display the results like this. So this shows just one of these sets, inflammatory response. And here this ridge plot shows the distribution of difference in expression values for all the genes in that set. Okay, so I know that things here can get a little bit complicated. And so before we get too wrapped up in the details, I want to take a brief interlude to provide some context about how to interpret these results. And one reason I like to take a pause here is because we actually know quite a bit about pox viruses. And that's thanks to a different pox virus that's been very important in human history. So think about this question for a second. What poxvirus has been incredibly important for humans? If you don't know, here is a picture to jog your mind. You might be thinking about chickenpox, especially if English is your first language. But in reality, what I'm thinking about is smallpox. And so this picture indeed shows smallpox, which was caused by a different pox virus, the variola virus. This, of course, was an incredible scourge of humanity. It caused a lot of death and sickness, especially among children. But it's also one of our greatest success stories. And that's because smallpox is no more. So let me tell you about our brief history with smallpox in 3 pictures. So at the end of the 1700s, a physician named Edward Jenner realized that he could inoculate people with a different pox virus, a cowpox virus called vaccinia. And exposure to vaccinia rendered people immune to smallpox. This, of course, is the basis of vaccination, which is something that continues to be quite important to us. And over the next 2 centuries, a concerted vaccination campaign was successful in eradicating smallpox from the face of the earth. This was an incredible achievement, one that we have done with no other human disease. But vaccinia, this cowpox virus, continues to be important to us. And that's because it's become an essential model virus in studies of host and virus evolution. And so there have been literal books written about vaccinia virus and pox virology. And we have learned so much from these laboratory studies. So on the basis of these studies, we know that certain things should happen when a host is infected with a pox virus. Specifically, there's various different processes that occur, starting with first and foremost, when the host detects a pox virus, it should sound the alarm. So the host vertebrates have certain viral sensing proteins that detect the viral incursion. They turn on interferon, which is our first line of defense against a viral infection. Interferon, in turn, spreads the word. So this protein triggers a signaling cascade that produces more intracellular signals. Some of these signals are transduced into the nucleus, where they change the cell's expression into an antiviral state. This in turn creates the expression of more signaling proteins, and these cytokines are exported out of the cell, where they can recruit leukocytes to the site of infection. They will activate our innate immune or our adaptive immune system, and this ultimately will trigger the production of antibodies and clear the infection. And if things are not going well, the host also has the option to sacrifice the cell to prevent it from being co-opted by the virus. And this, all of these different functions are very complicated, and I've sort of simplified to a great deal. These all help us to fight off viruses such as pox. On the other hand, what we've learned from these laboratory experiments is that the viruses have their own really fascinating and complex methods of immune evasion and host manipulation. And so at every step of these pathways, the virus can interfere with by obstructing signaling, interfering with interferon. The virus, of course, controls transcription and translation. That's the goal of the virus, to co-opt the cell's machinery. And even pox viruses even have the ability to inhibit apoptosis in some cases to continue preserving this viral factory that they've created. So indeed, much of the outcome of this disease may depend on whether the host or virus is more successful at controlling the cellular machinery. Okay, so with this little interlude into immunology behind us, let's go back to the finches. Here, I'm showing you again infected ground finches versus uninfected ground finches. And these are all of the hallmark sets that were significantly enriched in the infected birds. And so again, there's some esoteric acronyms in here. But when I look at this list, some things popped out at me. First of all, we saw a number of sets that were related to innate immunity. And so these correspond to these various processes that I mentioned, such as interferon, which is our first line of defense against viruses, other cell signaling sets. So these are all involved in immune signaling and spreading the word about infection, as well as sets that are signs of general immune activation, such as the inflammatory response. and even that cell sacrifice with interferon gamma and apoptosis. So apart from these immune sets, there were some other sets that seem to be associated with tissue repair, so that'd be coagulation, and sets that might be related to the fact that this virus causes lesions that need to be healed. But in the veggie finch, we see a slightly different picture. So first of all, we see a number of hallmark sets that are again involved in innate immunity, though overall the p-value support for enrichment of these sets is a little bit lower. We do see those sets that are involved in tissue repair. But what was fascinating to me about the vegetarian finch is that we also see a number of interesting sets that hint at potential viral manipulation and changes in the cellular function of this host. And so that includes a decrease in oxidative phosphorylation, which suggests that something is happening in infected vegetarian finches that is changing the cellular metabolism of these hosts. Another one that I'd like to highlight is the hallmark set cholesterol homeostasis. And so at first blush, this doesn't really have an obvious connection to viral infection, but it actually turns out that viruses care quite a bit about the lipid environment of their hosts. So of course, viruses have to enter into the cell. That involves passing through a lipid membrane. When viruses are replicating, they need energy as well as these building blocks to create more virons. And that requires lipid packaging for the virons, and that requires lipids as well. Oops, sorry. And so in general, we see through other experiments that pox viruses do care quite a bit about, in particular, the cholesterol environment of the host cell. And they have trouble replicating, in fact, when cholesterol is limited. So the fact that this hallmark set was upregulated in infected vegetarian finches suggests that perhaps this virus might be gaining a little bit of a better foothold in this host and is maybe a little bit more successful at manipulating the host than we see in the ground finch. So putting this together, what is the cellular response of birds to an emerging virus? So far, this experiment has given me signs that finches recognize and respond to a virus. And you know, in many ways, this is encouraging because we didn't know how the birds would respond to this virus since it's new. And there are many other cases in which there are other island species that are quite susceptible to infection. Something that was interesting that came out of these results as well is that we do see conservation in the antiviral response among vertebrates. And so these pathways that are upregulated in mice and in human cell lines, they actually turn out to be turned on as well in birds in a very different wild setting. And that shows us how laboratory studies and model organisms can help us understand wild ones and hopefully vice versa. But what we do see, which is interesting to me, is variation between species. And I'm not sure that I would have expected this because previous studies suggested that in many ways, Darwin's finches, which are fairly closely related, share a lot of immune genotypes. The previous studies suggested that they sort of behave as an undifferentiated single population, at least immunologically. But these differences between species suggest that there could be important variation among these birds and how they respond to this pox virus, and perhaps that some species are more susceptible than others. This is just a first pass, and so still to come in my research in the system is to follow up on some of these questions to understand first what underlies variation among species, why might some species be more susceptible than others. Then if it turns out that there are species of concern, to measure what are the effects of pox on individuals and populations. And this is work that I started just this past month in another trip to the Galapagos. And I definitely want to recognize that there's some limitations to this study. And so I've been thinking about different ways that I could validate these patterns and different approaches to studying pox infections in passerines. And that's something that I would love your suggestions on, or I'm happy to talk more about my ideas. So for now, I'm going to leave the pox virus to the side and switch gears a little bit to talking about the effects of parasites, not just on the internal function of the host, but the ultimate effects on their fitness and reproductive success. So here I'm going to tell you about the other parasite that's been a main focus of my research in the Galapagos. This is the avian vampire fly, also called Philornis downsi. This was the focus of my dissertation work at the University of Utah. So this is a parasitic fly that lays its eggs in the nests of birds. Those 3 larval stages that you see on the left-hand side, they feed on baby birds and their mothers, and they can cause a lot of nesting mortality. So indeed, we are worried about this. We're very worried about this fly in the Galapagos because it does cause very high mortality among Darwin's finch nestlings. So here you can see the pathology of parasitism. On the left-hand side, we have a little baby finch. It's non-parasitized. It's pretty ugly, but it's very healthy. It's large and rosy and well-fed. And on the right-hand side, we can see a heavily parasitized nestling. This nestling is small for its age. It's anemic. And you can even see the lesions on its abdomen and in its nares from where the larvae have been feeding on it. So when I came to this project, as terrible as these effects were on the finches, I was actually interested in a different species, the Galapagos mockingbird, also endemic. And this bird is fascinating because even though these finches were dying left and right, the Galapagos mockingbird seemed to be fairly tolerant to parasitism. And so here, I'd like to introduce and differentiate between 2 different strategies of host defense. So everything I talked about with pox viruses and our immune system falls under the category of resistance. And so that's the strategy where the host reduces its parasite burden, either by fighting off the parasites or removing its parasites, and that's how the host survives. Tolerance is a different strategy through which hosts are able to compensate for parasitism without reducing their parasite load. And so this is what we thought was happening in the mockingbirds, that even though they were heavily parasitized, many parasites in their nests, their nestlings were still fledging somewhat normally. And through behavioral observations, we determined that these mockingbirds appear to tolerate the vampire fly through increased provisioning. So parasitized nestlings would beg more, the parents would deliver more food, and supposedly they would recover this energy lost to parasitism. And in many ways, this defense seemed pretty tidy. You know, this is relies on behavioral plasticity of the hosts. It's not specific to this parasite, and it seems a good way for a new host to deal with this alien threat. However, this defense also presented something of a weakness for me, and that's because of the environment of the Galapagos. So let me show you what I mean. Most of the year, the field site where I work looks something like this. It's super dry, nothing looks like it's alive. Basically, the only things that we see to eat, or the only green things, are cacti. And so this is a pretty tough place to make a living if you're a bird. However, during the rainy season, which comes from about January to May, the landscape transforms. And so the rains come, there's a huge boom in growth, and that is followed by a boom in larval arthropods. And that's what the birds rely on to feed their young. But both the timing and the duration of the rains vary a lot from year to year. So in some years it's very rainy, and other years it's very dry. And it just turned out that the 2 years that we saw that mockingbirds were tolerant, and this was work led by my colleague Sarah Canuti, they happened to be pretty rainy. But those years were followed by 2 years of pretty severe drought. And we could see that both in the landscape and in sort of the mean amount of rainfall that was falling in each breeding season. And so this variation in weather gave me the opportunity to answer this question: is tolerance dependent on food availability? To do this work, I went back to the field and looked for mockingbird nests. They make these big old spiny nests in cacti and other spiny plants, everything spiny there. And so when the nestlings hatched, we would treat nests either with 1% permethrin, which kills P. downsi larvae, or with water, which allowed them to be naturally infested. We hung videos, our cameras, at a subset of nests so that we could record the behavior of mockingbird nestlings and their parents. So here you can see parents delivering some tasty snacks. We measured the growth and condition of the nestlings at 2 different time points. And then ultimately we determined fledging success by resiting color-banded fledglings. When the nestlings died or fledged, we would take down the nest. And here we could accurately count the number of parasites that were contained in each nest and directly relate that to the reproductive success of the parents. So right away, we noticed that in each year, parasitism takes a toll. So for example, here, this is hemoglobin concentration of the nestlings, where higher hemoglobin concentration is better and lower indicates anemia. We have these data from 3 years of the study. And we see in each year, those parasitized nestlings, those blue dots, have a lower hemoglobin concentration than the non-parasitized ones. However, the ability of nestlings to compensate for this damage varies among years. So here is the mass of nestlings when they were about 11 days old over all 4 years of the study. And in the first 2 years, which were our rainy years, there was just barely a difference, a non-significant difference in the mass between our 2 treatments. However, in the subsequent 2 years of the study, we saw a significant difference in the mass between non-parasitized and parasitized chicks. Ultimately, this resulted in differential fitness effects of parasitism and variation among years. So on the y-axis here is the percent fledging success for our 2 treatments across the study period. And we see that in the first 2 years of the study, again, there was no significant difference between treatments and reproductive success. These mockingbirds were completely tolerant to parasitism. Ultimately, it did not matter to these parents if their nests were parasitized or not. However, again, in these last 2 years of the study, there was a huge fitness effect of parasitism, and those nests that were parasitized fledged very few young. In terms of this idea of tolerance, let me show you a little bit about how we quantify it. So tolerance is usually depicted as the slope of the relationship between parasite abundance and fitness. And we can imagine 2 populations. So one whose fitness decreases really steeply with more parasitism, and another whose fitness stays relatively the same, even with more parasite abundance. And if we draw lines on these populations, we would say that the group with flat slope has a high tolerance, and the one with a negative slope would have more lower tolerance. And so these are our actual data. Here on the y-axis is percent fledging success, where each point is a nest. On the x-axis is parasite abundance in that nest. And we do see that in the first 2 years of the study, we had high tolerance. Even nests with more than 100 parasites were still fledging most of their offspring. That, however, was followed by 1 year of intermediate tolerance and 1 year of really low tolerance where most parasitized nests failed. And indeed, these correspond to the rainy and dry years of our study. So to try and connect these dots a little bit more about why tolerance was so low in the dry years, I turned to the videos and found that provisioning rates positively correlated with tolerance. And so years in which parents overall in the whole population spent more time feeding their offspring, we found that tolerance for the mockingbirds was higher that year. Putting this all together, it's clear that environmental conditions mediate tolerance. So rainfall really dictates so much in the Galapagos. It's tied to the growth of vegetation, which really dictates how much food is available to nesting passerines. And so in years in which there's abundant food availability, mockingbirds have plenty to feed their offspring, and they can recover the energy lost to the parasites. However, in years in which there is not so much food available, well, non-parasitized mockingbirds, they actually still do just fine, and they still fledge the same number of chicks. However, with the limited food availability, they cannot tolerate the extra burden of parasitism, and parasitism has a huge cost to reproductive success. And so in general, both of these studies have taught us a bit about how introduced parasites affect Galapagos birds. Both the POC study and this one demonstrate that naive hosts are not always defenseless. And so in particular, we see that these mockingbirds in many cases are just fine with this really virulent parasite. They can compensate for its effects when there's enough food in the environment. We also have learned about the mechanism of this particular the mechanism of defense in this case, and we see that behavioral plasticity is key to tolerance. And so in some ways, this is, like I said, a very tidy defense. The mockingbirds are able to respond flexibly to an additional stressor in their environment, but when environmental conditions allow. And so this is an example of how environmental conditions can really mediate host parasite outcomes. And this is important to understand because in many cases, we have a tendency to think of host parasite interactions as somewhat static, a parasite is virulent or it's not. But I do believe we have to think about these interactions in a more ecological context, especially in a changing environment. And so in an environment as variable as the Galapagos, which experiences huge swings due to El Nino or La Nina effects, we know that in the future, changes to this climate may have really drastic effects on the animals that live there, and they can mediate different outcomes between species, including between these mockingbirds and these introduced parasites. So wrapping things up today, I'm going to end by talking a little bit about the work that I do on the islands in community education and training. I might ask you, how many people live on the Galapagos? I think a lot of people who haven't been to the islands may not realize, but there's actually a growing population of people who are born and raised and live their whole lives in the islands. And in fact, there's some 30,000 people who live in the archipelago. Most of them live on the island where I do my work, Santa Cruz. And so I think that it's incredibly important for me as a scientist to work with the community to promote the sustainable future of these islands. So over the past few years, I have built a workshop program in collaboration with the Charles Darwin Foundation, and thanks to some support from National Geographic, to do workshops to share some of the principles that I use as a scientist and some of the results that I've learned about invasive parasites and pathogens of birds in the Galapagos. And the basic principles of this work are to improve access to science education, particularly for young people, to build capacity and interest in citizen science for the public at large, and indeed to support local management and conservation, because I recognize as a foreigner, as an American, I'm very privileged to work in these islands in this different country, which is not my own. And I think I do have a responsibility to support the ongoing work of institutions like the Galapagos National Park, the Charles Darwin Foundation, and all of the people who are living and working on these islands and working to manage their populations and support conservation. And so that's why one reason that one thing I think has been particularly successful in this program is that over time, more and more these workshop components are being led by local members of my field team. Here's pictured Diana and Hanai, who, like I mentioned, are born and raised in the Galapagos. They've learned to do avian field work by working with my team, and now they can share what they've learned and teach others through these workshops. And after a brief hiatus due to the pandemic, I was able to return to the islands last September, where I led two-day workshops in collaboration with the Charles Darwin Foundation and with the partnership of a number of different collaborating organizations. And these workshops were super fun. They combined lecture, activities, and practice in the field to talk about avian diversity in the Galapagos and bird conservation. Since I'm from the Cornell Lab, we of course employed some of our flagship citizen science programs and applications, such as eBird. And one thing that we took the opportunity to talk about is the use of hashtags to record certain notes that we might want to keep track of in these populations. Such as we promoted the use of a hashtag to document sick birds or birds that are infected with pox. And we hope that the use of citizen science could help us actually track the spread of these diseases through the archipelago and over time. And some of these programs are just super fantastic. If you're not familiar with Merlin, it's one that I use a lot because I'm not a great birder. Merlin has this functionality of being able to identify birds from a picture, or even now from sound. So the sound ID for the Western Palearctic, I think for Europe, was just recently released. So now you can sort of Shazam bird songs that you hear. And so I created little tutorials such as this video to show the functionality of these citizen science apps and to help people feel more comfortable using them. We also employed curriculum development, again, developed at the Cornell Lab of Ornithology, such as our K through 12 packet called Detectives de Aves, which provides different activities and discussion prompts to help children learn about their environment and about birds specifically. And then finally, I used pre and post workshop surveys to evaluate the effectiveness of this program. So one of the questions that I asked was, how comfortable are you with the following activities? And after the workshop, people reported that they felt more comfortable in all of these different activities for the most part. The only one where we didn't see a big change was in bird watching, which a lot of people were already comfortable with. And so these workshops did require a lot of work, and I want to specifically thank a number of my colleagues at the Lab of Ornithology who work in education, curriculum development, and assessment for their help in developing this curriculum. So looking forward, I'm really excited to continue this work, which combines education, population monitoring, and basic disease ecology to both understand how introduced species affect birds in the Galapagos and think about ways to mitigate their effects and conserve these really special birds. And I'll end today by talking about the themes of my work. So I've told you that hosts have various mechanisms of resisting or tolerating novel threats. They're not just, sitting ducks, as we might say. They in fact have ways to respond flexibly to a changing environment. But we do see that the outcomes of these interactions are in many cases mediated by the environment. So we need to take this ecology into account as we anticipate how animals will respond to a changing climate and why we might see the emergence of disease in unexpected places as environmental conditions change. And I do believe that addressing the effects of these invasive parasites requires education and requires collaborative work among conservationists, people working in education, and land managers. And so I just want to say thank you to a few people. And I want to particularly say thank you to Tere for this invitation. I didn't know where was the appropriate place to mention this because I didn't actually talk about the work that Tere and I have done together, but she and I met in Florida a few years ago when she was working as a postdoc there. And I came to learn a technique from her lab, and we just hit it off. We had a great time. She took great care of me and introduced me to this incredible group of Spaniards who she was hanging out with. And we just went out and I just had the best time in Florida, which is a very strange place, I will tell you, as an American. But in any case, I will stop there. And thank you so much for your attention. And I'm happy to take questions. Should I stop my share? I'll do that.
Hey, Sabrina, thanks so much. So we have Agus connecting in a second, and he'll lead the questions.
La verdad es que era para entrar Tere. Introduce Tere.
Yeah, actually, I had a question that I wrote on chat. I wasn't sure whether I should make it myself. It is about the first study. Well, first of all, thank you so much for the talk. It was really great. I learned.
Hello, everyone. So, okay, we're gonna allow Teresa to.
Okay, should I start? Yeah. Okay, I'll go ahead. I know that you said that this might be future work or already some ongoing work, but I wonder whether you have any ideas to whether the greater effect that the pox virus seems to have on the veggie finches, whether that translates into greater infection rates in the field. I don't know if you've looked at that or you have plans to do that.
Yes, that's definitely something that that I've looked at. Unfortunately, I've lost the precise numbers because my notebook was stolen, which is a terrible side story. Anecdotally, I can say that the veggie finches do seem to have higher infection rates and more severe infections. I don't have the precise data anymore, but that's something that we're looking at through ongoing population monitoring. So in my most recent trip to the Galapagos, which I came back this month, one of the projects that we were starting is almost a year-long monitoring census of pox of different species and continual capturing so that we can look at when are the periods of high pox prevalence, what environmental factors is pox correlated with, and hopefully begin to estimate the effects on the survival of different species.
Cool, thank you.
Should I take down my slides?
Sorry?
Oh, just wondering if I should take down my slides or if I should leave them up.
You can leave it like that. Thank you very much, Sabrina. This is a system that I have never given much of a thought. So it was amazing to see all these possible other possible diseases that could be awaiting for us humans from birds. But then that was it was very interesting because now we are so concerned about this. And there was one question though that aroused me. When you are doing these programs of conscientization about biodiversity, and probably you introduce things about the diseases and parasites that the animals have, how do you manage the reactions of the people? Like don't some people get a little bit they show some aversion or something like that? How do you manage this?
Oh, yeah, I think it depends on the person. I think sometimes people are so fascinated by gross things. So it's like they think, oh, it's gross, but then they're also looking closer. And so I think that's a very common reaction. I've been surprised, for instance, in these workshops, one that I didn't talk about is preparing specimens, so skinning birds for museum specimens. And in the Galapagos, we cannot kill birds intentionally, so we rely on salvages. And so birds that are hit by cars or birds that die in other ways, we can turn them into museum specimens. And salvage birds can be quite gross because they've been decomposing a little bit by the time you get to them. So I think it's pretty gross sticking your fingers in a bird, but the workshops, yeah, but it's interesting. And I was amazed that the participants in the workshops were so excited about it and so into it, because there's so few opportunities to actually see the inside of a Galapagos bird, since they are so well protected, which for good reason. But it's just an incredible opportunity to learn about anatomy, to learn about how a dead bird can still serve science. And so I found that in general, especially if I'm in the Galapagos, people are really interested in all aspects of avian biology, at least, the scientific community and other people who are self-selecting into this group, that as gross as these topics are, people are also just really fascinated by them.
It is really fascinating. I didn't know, in ectoderms, which is animals that I normally work with, they sometimes they show different behavior when they are parasites, concretely with respect to their thermal biology. I was wondering whether you made some interesting observations about that with the birds that you're studying.
That is also something I haven't thought about, but that's a great idea. We do see certain sickness behaviors in birds. So maybe they'll sit quietly, or maybe they'll lose a little bit of mass. But in some cases, it can be really hard to determine whether a bird is sick, because they often don't show many outward signs. But maybe we should start to take the temperature of birds when we catch them, and then we could, perhaps determine whether there's some sort of fever associated with viral infection, that sort of thing. It's just not something that I've done before.
Okay.
So I also have a question. I thought the part 2 of your talk on gene expression was really exciting. So now I'm going to ask, can you mute the We're getting a bit of an echo. So I was wondering about the gene expression part. So that's obviously you're looking at, I guess, phenotypic plasticity, right? You've got a control and a treatment. So where are you going to take that next? Are you going to look at local adaptation through SNPs, actual mutations in these relevant genes? Or are you going to look at transgenerational effects, or you know?
Yes, any all of that, if I can get some more money. So a few different directions. One would be to expand this comparison of gene expression effects to a broader number of species. So I have just 2 in this study, but you know, on the islands, there's 18 different species of finch. Some are on islands that have very low pox prevalence. Others are on islands where there's a lot of pox. And so I think there's some really fascinating comparisons to be made to determine whether, for instance, birds within the same clade, ground finches, do they respond in similar ways? And birds in different clades, do they have a different response? How much variation is there among this phylogeny in their phenotypic plasticity, as you say, to pox? And then from there, yes, I would love to know whether there are particular genes that underlie this difference in gene expression. So, you know, one thing that I've thought about, one direction to look would be in viral sensing proteins, because that's kind of one of the first levels of detection of the viruses are these suite of viral sensing proteins that tell the host that there's a virus present. And so, for instance, if these veggie finches have some sort of variation in their viral sensing proteins that makes them less likely to detect the virus, then that could be why they are not mounting a strong change in gene expression compared to the ground finches.
So what's from a methodological perspective, what would your approach be? Do you have any ideas? I mean, would it be like low coverage whole genome sequencing, exome capture? I don't know if you've given that.
It's tricky because it's a little bit like looking for a needle in a haystack. And so I've thought very carefully about, you know, there's a little bit of like, I could do a GWAS type approach, but I'd want to do a lot of individuals to make sure that it's relevant. You know, as we've learned from human GWASs, that a lot of times these results can be unsatisfying if we don't have like thousands of samples, which I don't. And so I think I've thought more realistically about perhaps looking at targeted candidate genes, so the suite of viral sensing genes, and using some sort of targeted sequencing approach to look at relevant immune genes rather than the whole genome.
Very cool. We have a question from the chat. Duarte Viana, our colleague, wants to know if would you tell us how long ago was pox introduced in the Galapagos?
Sure. We don't know the exact date, but from museum specimens, we know another paper guessed that it's been in the islands for about 100 years. So they have some samples that were collected a little more than 100 years ago that show signs of pox infection, and they were able to amplify pox from those specimens. But they think from specimens collected before that it's been about 100 years and not too much longer than that.
Interesting. All right. Does anybody else have any other question from the chat? No. Well, I think that's what the people have for today. The students are normally very, very shy, so please don't be shy. You can make your questions. It's okay. Well, then let's. If you wanna, do you want us to? We want to thank you very much for coming and give your talk here, Sabrina, and also Tere, to have you back again to make this presentation. Thank you very much to both of you, and to see you, everybody. Thanks all for coming and for attending this seminar, and see you all next week.
Thank you so much.